different when I registered for the conference. So plants engage in multiple interactions as members of a dynamic and complex community. These interactions take place both above and below ground. Some of the interactions, such as with pollinators and nitrogen-fixing rhizobia, are beneficial to the plant, while other interactions with root and shoot herbivores are antagonistic. Today I'll be presenting some preliminary data on herbivore interactions with native and invasive plant populations. When non-native plants are introduced to a new environment, they leave behind their co-evolved community members and interact with many new species. There are 3 ecological theories that explain, that hypothesize the role of plant-herbivore interactions in the success of invasive plants. The first, the enemy release hypothesis, states that native or non-native plants have left behind their co-evolved competitors and natural herbivores. So when they reach their new environment, they're able to thrive. The biotic resistance hypothesis states the opposite, that introduced plants are not well adapted to their environment, so native herbivores are able to limit their establishment and spread. And finally, there's the novel weapons hypothesis. This theory states that non-native plants in their natural environment have allelopathic chemical defense weapons that may not be as effective against their co-evolved community, but in their new environment, they are able to use the native herbivores are more susceptible to them. And then finally, there is the evolution of increased competitive ability. This is an evolutionary extension of the enemy release hypothesis, and it looks at the trade-off between growth and defense of chemical signaling that plants undergo. So it simply states that because non-native plants have left behind their natural herbivores, they can allocate more of their resources to growth, and less resources will be allocated towards defensive chemical compounds. We can also use these hypotheses to predict herbivore response according to them. So under the enemy release and novel weapons hypothesis, we would expect that native herbivores would prefer native plants, and that when they do feed on non-native plants, they would have lower growth rates. Conversely, under the biotic resistance hypothesis, native herbivores would prefer non-native plants and have higher growth rates. And from the evolution of the increased competitive ability, because non-native plants are now allocating more resources towards growth, they have a higher biomass, but this attracts more herbivores, and because there are less chemical compounds for defense, herbivores will have higher growth rates and do better on these non-native plants. So for this talk, the 2 questions I'm really interested in answering was, do native herbivores have a higher growth rate on exotic or invasive plant populations, and will they show a preference for invasive or native populations? To answer these questions, I used Medicago polymorpha. It's an herbaceous legume that is native to Western and Central Asia and countries around the Caribbean. It's also spread to the Americas, where it's considered an invasive plant. I chose the generalist herbivore, the soybean looper, which has a distribution within North through South America. For the rest of my talk, I'm going to refer to the native populations of Medicago as exotic because they do not have the evolutionary history with the soybean looper used 5 populations for each of exotic and invasive. So we wanted to know how well our soybean loopers will grow on intact plants. Will we see a difference in growth between our invasive and our exotic plants? So we used intact plants in containers, 5 plants per population, and we weighed larvae before and after our experiment to look for variation in growth. We're also interested in food preference. So for this, we originally had every invasive and every invasive exotic genotype paired with each other, but one just didn't work out. So we only have 19 pairs, and we have 5 replicants of each. Then we put an equal biomass of exotic and invasive leaf material onto a petri dish, and then measured the biomass again after the experiment. So what we found was that our insect growth did not vary between our exotic and invasive plants. In fact, it was highly insignificant when you compare the mean dry weight between our invasive and our exotic populations. However, when we then looked at the insect growth by genotype, we see that there is some genetic variation between and herbivore response. So this graph just has all of our populations on it and the dry mass, and is a matched letter between populations, which shows a significant relationship or significant difference between an insect growth. To determine if our herbivores prefer invasive or exotic plants, we required that 4 out of our 5 herbivores show a preference for one of the populations. This chart has arrows that point in the direction of the preferred plant population. In only 2 cases were our exotic genotypes favored. We scored these interactions, so if a population was preferred to receive 2 points and its partner received 0 points, there was a tie. Both received 1 point. When we ranked our herbivore preference, the highest score that you can receive is a 2. And it falls out that most of our herbivores prefer invasive plants over our exotics. So our early data suggests that this might be the enemy release hypothesis. Our loopers prefer to eat the invasive genotypes that they have some evolutionary history with, and they tend to have a higher growth rate on invasive foods as well. We're not really sure why this is yet, because when we look at measures such as digestibility, the rate of converting food to energy, we do not see any significant differences across the board. So to try and further answer these questions, our next step, so we're doing a much larger study with 40 genotypes. We're having 20 invasive populations and 20 native populations, so we can look for variation between our populations and also genetic differences. We're going to compare herbivorous response between both of our native and invasive, and also to a native clover that's found local to the California area. And then to finally get to the portion of that I thought I'd be presenting today, we will be introducing rhizobia so we can look at tritrophic interactions, because there are several studies that show that these interactions can have a huge effect on whether or not an invasive plant will become successfully established, and there's not a lot of data on that as of yet. And then finally, because we're really interested in just whether or not we're seeing rapid evolution between our native and invasive plants, we'll be using RNA sequencing for variation in gene expression. So I would like to thank the Freeze Lab at MSU, which I joined last fall, Beacon and NSF for funding. Thank you.
Do you know anything about the traits of plants that they use domestically?
Not yet. We do. There is a model of Ecological Theories, which so there have been some studies that looked at defensive signaling in those. So we are hoping that through our RNA-seq analysis, that we'll be able to determine what sort of defensive signaling and defenses that our polymorph is using. Sorry. We don't know yet what sort of defensive chemicals or signaling polymorph is using, but there are some studies in a related species, Stromcatula, that we're hoping to use as a model for any particular defensive compounds that polymorpha may have.
So you mentioned that you didn't find any differences in the digestibility indices. And so is the variance in growth of the larvae explained sufficiently by the variance in how much they ate and the consumption?
I think that it is, but I'm hoping that with this larger study that we'll be able to better parse out that our data will be a lot more significant and we can find some actual correlations.
When you do this larger study and you use rhizobia, where are you going to get the rhizobia from?
That was one of our sort of hang-ups. So we're actually splitting the study. The larger 40 are going to be from all over, but we're going to increase and focus only on rhizobia implant populations in Portugal and California, because we have rhizobia implants from both locations.
So I'm curious if what you know about the communities that the invasives are invading in California and Peru and wherever the other site was, and how closely related the species that are currently living or living continuously are, and to what extent they share the same natural enemies.
So there are, at least I can tell you a little bit about California. There are several species of both Medicago, the invasive, or also many Medicago, plus the native Trifolium in California. And we are less sure about what's actually eating them. There are studies that have not specifically looked at the potential herbivores, but just that they seem to be eaten by generalist anything that's there. Thank you.